Good morning. Today I want to walk you through something called microevolution and some of the evidence and theories surrounding microevolution. Microevolution is the changes in the allele frequencies or gene pools of a population over time. And so in order to sort of talk about this, there are a few terms I want to look at. Modern synthesis, I don't know if you've thought about this, but Darwin published his book in 1859. Wallace and Darwin co-published in 1858, and they really didn't know anything about genes at that particular time. Genetics took off at like kind of the beginning of or end of the 1800s and then all the way to mid-century. It wasn't until 1953 that we knew the structure of DNA. And so modern synthesis is really taking what we know about genetics and then blending it with Darwin's theories. Genetic variation occurs within populations, and this is what we say the variation that Darwin talked about was. We don't say that learned behaviors and traits like that can be like sort of acted on by natural selection because they're not always passed on, right, to the next generation. What we say is that natural selection acts on heritable traits or traits connected to genes. And so it really is the genetic variation in a population that is going to then change over time. Gene pools are all the alleles or the different forms of a gene in a population. Imagine this is not quite true, but imagine like you have eye color gene and you can have blue or green or hazel or light brown, medium brown, dark brown, almost black, right? These different colors, right, would be alleles or different forms of that eye color gene. And so gene pools represent all the different alleles possible in a population. And so remember, populations evolve, not individuals. So it's not the individuals changing over time, but it is the populations. Now, what is the source of new genetic variation within a population? The ultimate source of all new genetic variation is mutations. Mutations are changes to DNA. There are other sources where we mix up the genome, like in sexual recombination or crossing over, that we'll get to later on. But the ultimate source of anything new, a new trait, is a mutation, right? Changes in our DNA code. So microevolution then tracks the changes in these gene pools, the changes of these allele frequencies for a population over time to tell a story of how these genes are changing. Now, in the sort of early 1900s, there were a couple of scientists / mathematicians that studied evolution, and they wanted to kind of plot out what was happening in evolution. Hardy and Weinberg, that's the H and the W, published a theory called the Hardy-Weinberg equilibrium theory. This theory stated that given a number of assumptions, a population is not evolving if it is in Hardy-Weinberg equilibrium. This means that the allele frequencies are not changing for a specific trait. Remember, alleles are these different forms for a particular gene. Down here is their mathematical equation where P and Q are different alleles for a particular trait. So I want you to notice that Hardy and Weinberg, this equilibrium theory is kind of a null model stating when evolution or a population's alleles are not changing over time. Interestingly enough, they were kind of mathematically minded. So when they published this particular sort of mathematical equation, it came with a lot of assumptions, like this has to be true, this has to be true. And if all of these things are true, then the population is in Hardy-Weinberg equilibrium, then we can say that the allele frequencies for that trait are not changing over time. And so why this is interesting for a biologist is that these assumptions, when broken, gives us more mechanisms than just natural selection in order to have our populations change over time. Remember, Darwin and Wallace gave us only natural selection as a mechanism. Hardy-Weinberg give us some other mechanisms or ways, causes, things that can happen in a population that then cause the allele frequencies to change over time. So some of the things that have to be true for a population to be in Hardy-Weinberg equilibrium are the population has to be very large, right, in size, no gene flow is allowed, no mutations is allowed completely random mating and no natural selection. So in order to have that null model, no allele frequencies are changing or they're at equilibrium, they're not changing over time, you must meet these particular assumptions. So the interesting thing is if we break them, if we break these assumptions, then we can get allele frequencies changing over time, and we can get this and then have some new mechanisms for sort of evolution. So starting with number one, you have to have a very large population to keep that Hardy-Weinberg equilibrium. So what happens if we have a small population? How can we break that particular mechanism? So one way we break it in a small population is something called genetic drift. And this is the evolutionary mechanism where a population's gene pool changes due to random chance. I have these flowers over here, and if you have a very small population and you were to kind of like randomly draw out who gets to mate, right? Let's say we drew out these circled ones here. We're starting with an allele frequency for red of 70% and white 30%. If we only have these 5 individuals mate, then we get this second population. And look, our amount of red alleles to white have changed to be 50-50. If we only then selected 2 of those, right, small population again to mate, but look, they both have that capital R or red alleles, then 100% of our next generation would carry only red, and we've lost white, that white allele, completely out of our population. So this is an idea of how random chance, how small numbers can cause a population's allele frequencies to change over time. An analogy of this is if I asked you to flip a coin. If I had you flip that coin only 5 times or 10 times, right, you would probably more likely get something that is different than 50% heads and 50% tails than if I asked you to flip that coin 1,000,000 times, right? The larger the flip of that coin, then the more likely it's going to be about 50% heads and 50% tails. The smaller number of flips, like 5, means that you could get all 5 heads, right? Or all 5 tails. And if you did that, you would lose an allele altogether. Genetic drift can be caused by a couple of different things. One thing that can influence quite quickly a set of alleles in a population is a bottleneck effect or a near extinction event. Sometimes when things nearly go extinct, then our original population, if only a small number survive, it's almost like you're tipping the alleles through a bottle and you get a different kind of allele set at the end because you have a very small number left after that extinction event. And so we can get our alleles changing over time. Another way we see this is through founder effects. This particular island was settled by only, I think, a dozen individuals. And those dozen, one of the kinds of problems that one of those members carried was an eye problem that caused degeneration and sort of middle age to late age blindness. This island still today has a higher percentage of that kind of blindness than any other place in the world. Because so few people founded the area, they carried with them a very small number of alleles that got perpetuated in the future generations of that particular group. And we see that the allele frequencies for this island are still different, right, than the neighboring sort of countries around it. Now, Hardy-Weinberg equilibrium says that you cannot have gene flow. Gene flow is when a population gains or loses a fertile individual from emigration or immigration, right? So going into or out of a population. Anytime those fertile individuals move their genes into a population or away from a population, then the genes within that population change because of that sort of movement of that fertile individual. So if you have gene flow, if you have members that are like sort of leaving a population, this happens a lot in males, like in elephants, the males will be kicked out of the maternal herd and have to go find their own populations, right? Then you get movement of these particular individuals and changes in the allele frequencies. Hardy-Weinberg says that you cannot have mutations. Mutations permanently change the alleles possible for a population. And notice that this is really hard, right? right? Because like we're around mutagens all the time, UV light, sunlight, right? Things of that nature, and different chemicals are constantly surrounding us and then mutating our DNA or causing hits in our DNA. So this is quite difficult to control. To be in Hardy-Weinberg equilibrium, you have to have completely random mating. That means no selection of mates at all. So no mate selection, also known as sexual selection. This is an assumption that violates then Hardy-Weinberg equilibrium. And why does it violate Hardy-Weinberg? It's because if you have mate selection, then this means that certain individuals are getting to mate more, produce more offspring, and leave more of their genes to the future generation. This happens in birds like this bowerbird has set up this beautiful nest. The females come through, the males set up these elaborate nests, and then the females come through and choose. And it means that certain males might be mating more than others, right? Based on the nest that they are presenting. And lastly, the last break or way that we sort of break Hardy-Weinberg is to allow natural selection. So anytime we have differential survival, certain individuals surviving better, reproducing more, passing more of their genes on to the next generation, then you will have gene pools change over time. And this is a way to get sort of a break of Hardy-Weinberg equilibrium, or where allele frequencies are changing over time. Types of selection. So there are different sort of results of selection. You can have directional selection and natural selection. This is where you select an extreme form over another extreme form. So imagine mice could come in a black variety, a gray or medium color, and then a white variety. If you are selecting kind of a black morph over a white, so one of the extremes, then you would have directional selection where you're moving towards that particular direction. This might be because of camouflage or coverage or rocks and things like that, and how things are hiding or behaving within their environment. Disruptive selection is where you select the two opposite extremes. So this is where you get kind of a double hump where you're selecting black and white and selecting against gray, or that like intermediate morph. And lastly, stabilizing selection is where you select that intermediate. So then you're selecting for the gray mouse over the black and white. Stabilizing selection happens a lot. It's very common. This is things like birth weight in humans, right? If you have too big of a baby or too small birth weight, then that can like challenge either the mother's delivery if it's too large, or the baby's success at like sort of living if it's too little. And so we select an average birth weight as best or easiest. And so most babies are around 6-7 pounds when they're born, because it takes the pressure off these other two extremes that have some problems with them. So in all of these ways, we break Hardy-Weinberg equilibrium. So remember, Hardy-Weinberg equilibrium, for a population to not be evolving, that means their allele frequencies are not changing over time. And we say that that population is in Hardy-Weinberg equilibrium. In order to have this, you have to have these assumptions: very large population, no gene flow, no mutations, random mating, and no natural selection. So this is interesting for a biologist, because now from Darwin forward, right, we have some more mechanisms. If we break any of the Hardy-Weinberg equilibrium assumptions, then we get these other mechanisms causing microevolution, or allele frequencies in a population to change over time for a particular trait. And so if we get small populations, we can have genetic drift, or if we get gene flow mutations, sexual selection or mate selection or natural selection, then we break Hardy-Weinberg equilibrium and we can have some other mechanisms for evolution. I will see you next time.